For today's session, I was just thinking about explaining some dumb things in drug development to a dummy like me. For example, people say this word marker all the time, and it's so common in drug development and biology, but how would you explain marker to a dum-dum?
Okay, first of all, I don't agree with dummy. Okay. And with that, I'll explain what a biomarker is. This is easy, right? Because I have a third grader who asked me the same question. And this is
Uh,
what I told him. I said, just like how in your car, you have a fuel gauge or a check engine light or a pressure gauge for your tires, just like that for your human body, you have measurements that you can take from the blood or tissue that will tell you what is happening within, what the drug is
mhm.
doing to the body, or how is the drug targeting.
Maybe translate that into financial terms, which is like time or money for a drug developer. How does that save time or money for a drug developer knowing that kind of stuff?
For cancer, as you know, it's not a one disease. Every patient is
Mm,
different. Just like how our fingerprints are different, every patient has a different immunology or immune system. Biomarkers in drug development is to tell us whether a drug is targeting the right population. Most of our cancer drugs right now does not treat cancers in 100% of the population.
mm,
Biomarkers help us identify what actual populations does it treat. So this way
mm,
we can avoid wasting time and money and
mhm.
only treat patients who are going to benefit from it. That's the goal for biomarkers.
The order of magnitude with the public stats on oncology trials, it's like $100,000+ per patient to enroll, and it takes months also. The value would be to potentially reduce your enrollment down to a much more manageable number. Instead of a $20 million trial, maybe like $15 million, and then also go faster in terms of speed.
Yes, right. Yes. So the
Hmm.
money story is that it is to make
Yeah.
sure that the biotech who's investing in these clinical trials are not just throwing it out there and hoping something will stick, right? It is to ensure that they can actually treat the patient and that there is efficacy in the trial. A drug
Hmm.
can be a go or no go just because of the data that comes out of these clinical trials. And you want to
Hmm.
ensure that as a biotech or a sponsor for the clinical trials, you want to ensure that you're only treating patients who are potentially going to benefit from it. So that's the
Hmm.
money angle. The
Hmm.
angle for the patients is that when you're going into the trial, you have some hope, right? You're hoping that you're
Yeah.
going to be cured. So
Yeah.
for a scientist like me, that's a huge angle. You're going in hoping
Yeah.
you're going to be cured.
Yeah.
And if I can tell them that these are the markers in your blood that potentially are going to tell me that you're going to be treated, then it's a game changer, right? So that's how I see
Hmm.
it for the patients.
In terms of from the patient's perspective, number one thing is you're not doing the trials for fun. You want to get
Mhm,
cured of your disease,
mhm.
and showing these markers going up or down might mean that you're have a better chance of
Yeah.
actually getting cured. What are the common reasons patients don't go into trials if they have the option?
There are multiple factors, right? So before the trials were simple, I would say, compared to what we have now.
Yeah.
We have technology that has advanced so much that we can do multiple things with a few microliters of blood. This
Mm.
includes proteomics, transcriptomics, and you know the deal, right? You are in that space.
Yeah. Yeah.
So, because we have that much ability to identify things from blood or other samples, we extend what we can do when we
Mm.
design clinical trials, right? So we are
Hmm.
asking for, can the patient come in and get the infusion, which is about 4 hours long, and give
Mm.
blood every 30 minutes and give urine sample at the end of it. And it keeps going. And as a
Mm.
patient, imagine sitting there at the infusion center and getting the infusion, and you just want to go home, but you're being
Yeah.
told you have to give more samples. So
Mm.
at least where I'm working now, we are always trained to think from the patient perspective. If you were a patient, would you be willing to go in and get on this trial with the hope that it will
Yeah.
either help you or help someone who is struggling in the future
Yeah.
with the same
Yeah.
disease? And also the location, right? The location of the clinical sites. Not all clinical sites can do these intense trials.
Yeah.
There are only certain sites that can do it, which means that again, we are making these patients come in every 3 days or every few weeks to ensure they
Hmm.
get all the biomarker samples. meaning that they cannot go home. They might probably have to stay closer to the clinical site. So
Mm.
some of these things we don't appreciate as much while we're designing clinical trials, but it would be much better if we can take that into consideration.
So that's kind of like the translational gap, right?
Yeah,
Which is something that would be theoretically awesome, but then has to be confronted with the reality of
exactly.
a busy medical practice.
Exactly. Yeah.
You said the clinical trial sites that people have to travel for, but why isn't it just easier to go to a local doctor's office to do a trial?
Okay, so multiple things, right? So when we say a clinical trial, we for an oncology trial, we are collecting many blood samples to answer
Yeah.
multiple questions, which would mean that sometimes you need the staff to collect
Mm,
samples every few hours. Sometimes, based
mm,
on when the patient is coming in, this might go
mm,
into the night. Not
mm,
every clinical site or community sites have the ability to do that. You can only
mm,
do these in bigger academic sites like MD Anderson or MLE,
mm,
or where you have the ability to say, okay, even if this goes beyond the time of 8 to 5, I still have staff who can do it. As a biotech or a sponsor, when you're reaching out to
mm,
sites and you want to do clinical trials, you naturally go to bigger sites because you assume they can do what you're asking them to do,
mm,
and not
mm,
go to community sites because you're afraid that they might not have the infrastructure to do it.
mm. We hear from developers all the time that this is a very manual, tedious process. Seems like ripe for AI to speed this up.
Yeah, exactly. That's something I've always been thinking about for the past few months. There's a lot AI can do in this space. I
Yeah.
have seen clinical coordinators running around to get their job done, and it's a thankless job, right? Because they miss
Mm,
their lunch, they run to get their samples delivered to FedEx because the FedEx timeline for pickup was 4:30, but then they missed it because the patient came at 10 a.m. in the morning and the last sample had to
mm,
be collected at 4:30. So what do?
mm,
They take it, they run to FedEx to drop it off. There's so many components when it comes to clinical trials, right? So I think
mm,
that's the major challenge in making
mm.
it more effective.
Let's go back to your example of the missing the FedEx thing. It feels like Amazon has solved these problems in the 2000s. So,
Mhm.
what makes it complicated today? Amazon makes logistics look so easy. Obviously, there's a million engineers and people
Mhm.
behind the scenes making that happen. But if people know reasonably what the FedEx pickup is, why is that a challenge today?
Things happen, right? During a
Yeah.
clinical trial, things happen. The patient sometimes is not responding well to the treatment. It has to be stopped.
Mm,
You have to call the PI. You have to ask them what are the other drugs you can give them to mitigate that issue. Things don't go as planned. I wish there was an
mm,
ideal trial, but it never goes that way, right?
mm,
Because of these factors, it ends up being that we are collecting samples that goes beyond the window, resulting in a
mm,
deviation, which also adds up to the fact that you're collecting samples which cannot be delivered at the right time. For PPMC samples, you want them to be collected the same day and shipped out the same day for processing. I know you have a different experience there, but in general,
yeah, yeah,
in general,
yeah,
that's the goal. And
yeah,
I'll tell you what, Ramji. So when I was a preclinical scientist designing experiments for mice, I
yeah,
would get stressed out designing for 20 mice, something I can control, right?
yeah.
And these 20 mice, I plan 3 days ahead of time and I mobilize my entire team. I'm like, you do this, you do that, I'll do this. We're getting everything done, but never is it 100%, right? We
Uh huh,
always
uh
miss
huh.
something. If that's the case
Mm.
for preclinical studies, I cannot even imagine what it's
Uh,
going to be for clinical samples.
in the lab, it's interesting. If you look at the statistical distribution of errors,
Uh-huh.
the vast majority are what's called pre-analytical before a specimen ever hits a lab.
Hmm.
So, stability window, errors on requisition forms.
Hmm.
This is like 30% of errors. That leads to sample swaps downstream. Very little physical stuff are the source of errors. You mentioned biomarkers. How would you draw the distinction for, again, not a dum dum, but like a layman in terms of pharmacodynamics and biomarkers? How would you make a distinction, if any, between those?
Well, we use biomarkers to look at the pharmacodynamics of a drug. What that means
Mm-hmm.
is biomarkers are the signals, and the
Mm-hmm.
pharmacodynamics is the interpretation of those signals to understand
Mm-hmm.
how a drug is working, right?
Mm-hmm.
They go hand in hand.
Any companies you think that, besides your own, that you think are doing this really well in terms of interpreting all these signals, putting them together?
We have been doing that well for a long time. Most big pharma does a pretty good job in their PD analysis.
Mm-hmm.
And any drug that has gone from phase 2 to phase 3, I'm assuming, has done it right. The questions that we have asked now has changed. We want more answers. We want to know not just does this drug work, which was the case a few years ago. We also want
Yeah.
to know what else can I do to the drug to make it work? What is the combination that will help it? Which patients won't work? So we are making these
Mm.
questions even more challenging, making the whole trial, extremely intense.
But yeah, and also it seems like dosing is also more of a hot topic with Optimus.
Yes, Project
Yeah.
Optimist. FDA wants you to have two doses tested at the same time. I see why they want you to do that, but that also means that the biotech has to spend to get the right dose, right?
So it's 2x the cost, right?
Yeah. Before we used to guess a dose, we would have, let's say, 500 and then 800. We would say, okay, maybe the 500 hits the target, but 800 could be better. But now we know for a fact, no, 800 is not working. It's toxic. Let's just go with 500, right?
Mm,
So
mm.
I think Optimus helps us with that.
One of the things that we're seeing more is companies look at Turkey, Australia for clinical trial sites. Is that something that you're seeing?
Hearing
Okay.
a lot about Australia too these days. Before we said, let's do clinical trials in the US, but it became challenging because every
Uh,
biotech out there was going to the same set of clinical sites to make these things happen. They were
yeah,
stretched thin, they couldn't take more trials. So then we said we'll do global trials, right?
yeah,
We do APAC, China, Singapore. We just expanded, which
yeah,
also adds to its own confusion, right?
yeah.
You're having issues doing all these things in one place. Imagine what it would be for different countries. But now FDA is again saying something else. It's like, hey, you're treating US patients, so you should have majority of your patients from US, right?
Mm-hmm.
So it's going back and forth. Before we were okay with
Mm,
global, but now they want more patients from the US to be recruited on your trials because that's your target.
mm. Because of the demographics of the US.
Yeah, exactly. I mean, a lot of biotechs do Europe, Australia, they do global studies, but there is a catch in terms of operational feasibility and also in terms of the cost. But something else to note is that some diseases, for instance, some of the rare diseases that are rare here in the US, are more prone or more common in other countries. So if you
Mm,
want to do a rare disease, you would potentially go to a country where it's more common, just so your recruitment is
mm.
faster.
So I guess turning back to your journey, why did you get into drug development?
From my accent and from my name, you probably know I'm from India, right?
Hmm.
So
I guess.
back in India, when I was growing up, we were asked to decide what we wanted to do when we entered our
Hmm.
10th grade. We had to decide our life for the next few years. I didn't know
Hmm.
what my life was going to be, but I knew for a fact that I loved biology. I
Mm,
loved immunology. I was in awe of how the human body can fight diseases. When my professor told me in undergrad that if you have an infection and then 10 years later, if you have the same thing, your body knows how to detect it. It would mobilize
hmm.
the army against it. I was like, what? It can remember? I did my undergrad in biomedical engineering and then moved here,
Mm,
and there was no looking back after that. It's just
mm.
the love for cancer immunology. That's it.
That's incredible because I feel like biology in the US is taught as this incredibly boring
Ah.
discipline, and it sounds like you actually learned it. It was just like a bunch of memorizing, oh, the Golgi apparatus and the
Oh,
mitochondria is a
really?
powerhouse.
Oh, well, it was. I think I was just a nerd. How about that? Because
But how did you get beyond memorizing then?
I had some professors or teachers who were extremely inspirational. They
Mm,
made it into more of a fun experiment rather than
yes.
memorizing. And I'm not good at memorizing, I'll tell you that.
Mm,
But if you can tell me why something is some way, then I can understand it better. And
yeah,
immunology is a language by itself, right?
it really is. Yeah.
Exactly. It humbles you because every time you
Oh,
learn something new, and at the same
yeah.
time, you feel like you know a lot about the human body. So
Yeah.
that's the fun part of it.
Well, thanks for coming on.
Yeah.
This has been awesome.